Our today's class starts with a question. What is genome? So genome is an complete set of genetic material or the hereditary information. It's a complete set of genetic material. Like in human, we have 23 pair of chromosomes. Whatever the genome or whatever the gene is present in this 23 pair of chromosome is termed as the genome of human. So it is encoded either DNA or for many type of viruses, it is RNA. So mainly for the eukaryotic and prokaryotic organism, the genome is consists of the DNA. And in case of some viruses, it consists of RNA, like the influenza virus, like the HIV virus. So the genome includes both gene, which are the coding sequence, and the non-coding sequences of the DNA. So genome includes both of these two parts of the genetic material. So genome is thus the entire collection of gene and all other functional and non-functional DNA sequences in an organism in a haploid set of chromosome. In a haploid set of chromosome. So we need to consider 23, that is the one set of chromosome of human. So that includes structural gene, regulatory gene, and non-functional nucleotide sequence that we are generally termed as non-coding sequences. So what are the structural genes? Structural genes consist of DNA segments that code for some specific RNA or proteins. So it can code for the specific RNAs like mRNA or rRNA or tRNA, etc. So whatever the different kind of RNAs are, or some other kinds of RNA are also present, and proteins. So whatever proteins is encoded, that is a part of the structural gene. So that encodes mRNA, tRNA, snRNA, cRNA, whatever the RNAs are present, that is the structural part of the gene. Now comes to the functional sequences. So as you people already have some idea about the transcription and transcription part. So for the transcription, we need some regulatory sequence that is the promoter or the operator or the initiation site of the transcription. So these all are, so these are mentioned here. So these all are comes under the functional part. So these are required for the functionality of the DNA or functionality of the chromosome. So now comes the non-functional sequences, which mainly includes introns, repetitive sequences. So these are need for coding, regulation, and replication of the DNA. So much more in basically they are not that much involved in the structural or functional part. They have some other roles in the evolutionary perspective. So in some other classes we will discuss about the non-functional sequences, but these three are mainly the parts of the overall basis. These three are the main constituents of the genome structure. So genome of all living organisms represents their hereditary material and is formed of DNA. So whatever the hereditary material a particular organism have, mainly we will discuss about prokaryotic and eukaryotic organisms. We will not discuss about the viruses here. We will discuss in some other classes about the virus genetic material. So in prokaryotic cell, genomic DNA forms a single circular chromosome without basic protein, lies in the cell cytoplasm in the nucleoid region. So as we already have the idea of prokaryotic cell structure, and we know that prokaryotic cell is basically devoid of the true nucleus part, and that is why it is called the nucleoid structure or nucleoid region. So prokaryotic cell is basically composed of a single circular DNA, which we also call the single circular chromosome, and that DNA is not basically bound with the proteins. So DNA protein complex is not present here, and genomic DNA. So the genome is basically composed of their single circular chromosome. But in eukaryotic cell, DNA is associated with some basic proteins. We call it histone proteins. We will know about its structure in later in this class. So it forms a long chromatin fiber, and chromatin fiber forms a network. So it's a different stages of network which are formed in case of the eukaryotic organism, and it enclosed in double-layered nuclear envelope. So eukaryotic cell actually have a true nucleus, and the true nucleus is basically composed of some parts which includes nuclear membrane, nuclear core, nuclear material, nucleolus. So these are the basic parts of a true nucleus. So, and these eukaryotic cells have its DNA associated with the or binds with the histone proteins, which is present in the inside the nuclear envelope, and that condensed into chromosome during cell division. Chromosome is a specific structure which is actually visible during the time of cell division. Before that, we don't actually term this structure or this DNA structure as a chromosome. We call it a genetic material or nucleoid structure. So if we consider that the genome size of different organisms, we will find in human, the total size of the genetic material is 3.2 billion base pair. And the total gene number is approximately 25,000. Initially, it was at the very beginning of the before the beginning of the this genomic or this genetic study era. It was basically thought that human have 1 lakh number of genes, but now after the Human Genome Project, we know that we have more or less 25,000 of genes. So average gene density is the 1 gene per 1 lakh basis. The total chromosome number is 40. In case of musculus, that is a mouse, they have a 2.6 billion of base pair of genome, which also include 25,000 of gene, and they have 14. So in this way, uh, the Escherichia coli, they have 4.6 million of the total base pair. So that is quite less in number. Overall, 3,200 of gene, and 1 gene per 1,400 bases. So total one, we do not call it a chromosome. The circular chromosome is present in case of the Escherichia coli. So, in eukaryotic genome, the nucleus is the heart of the cell, which serve as the main distinguishing feature of the eukaryotic cells. It is in an organelle, with actually the nucleus is an organelle which is actually present inside the cytoplasm, and the nucleus itself have a jelly-like substances or a liquid material inside it, and we call it a nucleoplasm. And if we combine the nucleoplasm and the cytoplasm, we will get protoplasm. The nucleus mainly contain the nucleolus and the thread-like coil structure, which remain actually suspended in the nucleoplasm, which also known as the chromatin structure. So if we consider chromatin, chromatin is a complex combination of DNA and protein that makes up the chromosome, which is very important part. And we are going to discuss a structural part of also. And the major protein of the chromatins are the histone proteins. There are 5 histone proteins which is involved in the development of this chromatin structure. The function of chromatin is to package DNA into the smaller volume to fit in the cell and to strengthen the DNA to allow mitosis and meiosis and to serve as a mechanism to control gene expression and DNA replication. So these DNA protein complex have a several role which includes its compact packaging, because if we are able to unpack the DNA material, it is quite huge, it is quite large, and it is very the cell need a very complex or a complex mechanism in a simpler way to make this chromosome so compact that it can be easily fit inside the nucleus. So this is the somehow the structures of the chromatin and chromosome. So this is the chromatin structure. This is some uncondensed chromatin structure which is present inside the chromosome, and this chromatin structure is in the latter part is getting condensed, and after that so this is the chromatin structure. This is the DNA. These are the histone proteins. Yellow, this blue and green are the histone proteins. These histone proteins are complex with the chromatin structure to form this kind of a histone chromatin complex, histone DNA complex, and they are again complex to form the solenoid chromatin fiber structure. And after that, they form these chromosome, overall this typical structure of the chromosome. And in chromosome have several parts, and which include the centromere part, which is the main constriction here, the arm, and also the telomere region. Telomere region is the end region of the chromosome. We will actually discuss this telomere region later. So, if we are going to study the organization of chromatin, we have to start from a resting cell, a cell which is present in its in its resting phase. So a cell in its resting phase, that means a cell particularly in a non-dividing phase. The genome is in the form of nucleoprotein complex and the chromatin. So this nucleoprotein complex is called the chromatin. So the information stored in DNA is organized, replicated, and read with the help of variety of DNA binding protein. So that includes the structural protein. Histone is the main structural protein which helps in the DNA packaging. They are main structural protein. They have low molecular weight. They are very conserved. They have a very conserved sequence in case of the evolution. So in case of the eukaryotic organism, the whatever the evolutionary parts are, but this histone is very conserved. The change among the different species is very less. So these basic proteins with high proportion of positive charged amino acid, as they have a positive charge amino acid and DNA is composed of nucleic acid, they have a negative charge, so they are able to bind with each other. They bind to DNA along most of its length. The positive charge helps histone to bind DNA and play a crucial role in the packaging of long DNA molecule. So these histone proteins are the main part of their main function is to bind the DNA, bind with the DNA, and helps in DNA compaction or packaging. Now, this comes the functional protein. Those are the non-histone protein. They are basically associated with the gene regulation. So, in some cases, in different when different genes are need to be expressed, so these proteins are actually involved in the gene regulation part and their functional part. So now we need to know what is the nucleosome structure. Nucleosome are condensed several times to form the intact chromatids. So nucleosome structure starts with the DNA and it's binding with the histone protein. So the histone proteins, if we consider the histone proteins, so histone proteins is basically made up of 5 different proteins, that is H1, H2A, H2B, H3, and H4. Among that, H2A, H2A, H2B, H3, and H4, they form a tetramer structure. They form a tetramer structure, and this tetramer structure is helps in the packaging of the DNA. And the histone 1 H1 protein is basically present within that the two hexamer structure. That means the octamer of this protein of this H2, H2B, H2, and H4 is present, and H1 protein, H1 histone protein is basically the connector protein. They are They are basically able to connect the two octamer structure and the wrap up with the DNA. So the chemical composition of the chromatin is DNA, that is 20 to 40%, and they are the most important constituents of the chromatin. RNA, that is associated with the chromatin as ribosomal RNA, mRNA, or tRNA, and rest of the parts are the proteins, that is composed of 55 to 60% of the associated chromatin. So histone is a very basic protein. It is mainly constituted of 60% of the total protein, almost 1:1 ratio with the DNA. So there are 20% of the non-histone proteins is also present, which is basically not that much important. We will discuss something later, and that includes the actin protein, L and B tubulin, myosin. These are the different contractile proteins which involve in the functioning of the chromosome condensation, and they also basically they involve in the chromosomal movement during the mitosis and the meiosis process. And this 50% include all enzymes and cofactors which involve in the replication, transcription, and regulation of the transcription. So now, if we consider the ultrastructure, so this chromatin structure is a multi-strand model, or we can consider it into the folded fiber model. But the most important model, most recognized model is the nucleosome model, which is widely accepted. And this nucleosome model, it is stated that DNA is tightly bound to the histone protein, which serve to form the repeating array of DNA protein particle called the nucleosome. We also call it a strings on bead structure. String on bead structure. So strings. So these are the histone beads in form of octamer. Histone beads in form of octamer, and DNA is wrapped into that bead. So DNA is the string, and the beads are the histone. So DNA wrapping around the nucleosomes. The 30 nanometer condensed chromatin fiber consisting of nucleosome array in their most compact form that we call the solenoid structure. So 30 nanometer structure is called the solenoid structure. The higher level of packing into the metaphase chromosome that consists of loops, domains, and the scaffold structure. We will come into that. So this is the bead on a string. Bead on a string. That is, these are the octamer, histone octamer. This is the histone 1 protein. This is the histone 1 H1 protein, which is actually a linked protein between two beads. And this is the DNA. This is the DNA which is wrapped around the histone, wrapped around the histone octamer. So overall, this is a 55 Armstrong nanometer structure, 55 Armstrong, and in its height and in its length, it is 1110 nanometer structure. So overall, roughly, it's a 10 to 11 nanometer structure. So this nucleosome structure includes the two copies of the H2A, H3, and H4. That is why it is called an octamer, histone octamer. Two copies of histone H2A, H2B, and H3 and H4 is present. 147 base pair of DNA is wrapped around the nucleosome. So whatever the DNA is wrapped, if you count its base, it's a 147 bases of DNA is wrapped around the nucleosome. Tails emit from the core, some nucleosomes contain histone variants. So some nucleosomes, there are various less variants are available, but then also H1 is the linker histone. H1 is not present within the octamer structure. They are actually the linked chromosome, linked histone. So this is the hierarchy model of the DNA structure or the nucleoid structure. So that is the first, it starts with the DNA double helix, which is the 2 nanometer structure. Then it's a nucleosome structure, that is a beads on a string. So these are the beads and these are the string. This is the string. So this is a 11 nanometer structure. After that, it comes to the 30 nanometer structure. So when six of these individual beads comes together, so 6 bead comes together. 3, 4, 5, 6. 6 beads comes together to form the particular condense 30 nanometer structure. 6 beads comes together to form these kinds of a structure, which is called the 30 nanometer chromatin fiber structure. And after that, it forms a scaffold structure. Scaffold. That means this 30 nanometer structure forms some loops. This loops is associated with the chromatin loop domain. That is the 300 nanometer structure. And after that, that forms the higher order packaging of the chromatin loop. Again, it forms some chromatin loop. And finally, it condensed into the metaphase chromosome, which is a 1400 nanometer structure. And this is actually visible. That is a 1.4 micron. This is actually visible under the microscope. And you probably have done some practical, or you probably will going to have some practical in order to visualize this metaphase chromosome using onion root tip as a model. So now we need to know what are the different kinds of chromatin which is present. So chromatin, if we consider chromatin, we can easily divide this chromatin into 2 parts: euchromatin and heterochromatin. So euchromatin is a lightly packed form of chromatin that is rich in gene concentration. So chromatin, that's a true, means true, this true chromatin part. That means the main gene is present in this part. That is the most active part of the chromosome. Most active part. This part is most active part. That means this part is basically more active in terms of the transcription and translation process. So transcription, not translation, transcription process. So it takes up light chain and represent most of the chromatin that disperse after mitosis has completed. Consists of structural genes which replicate, transcribe during G1 and S phase and in the interphase. So whatever the main genes, whatever the main structural or functional genes that are present in this eukaryotic, sorry, euchromatin part. So this is considered genetically active region. So since it has a role in their phenotypic expression of the gene, so if we are able to stain DNA, we may get some band. Some part represent as a dark region, some parts represent as a light region. Dark regions are the heterochromatin and the light regions are the euchromatin. So during metaphase, it takes up the dark state. Heterochromatin. Heterochromatin is a tightly packed form of chromatin that takes up deep stain during interphase and prophase, but metaphase takes up light stain. So it's just the opposite. In the euchromatin part, they are actually much more involved during the G and S and the G2 phase. That means in the interphase. So that is why if we try to stain these euchromatin in the interface portion, they will take a light stain. And if you just try to stain them in the mitotic in the during the time of uh, metaphase, it will take a dark stain. But euchromatin is just the opposite. They will take dark stain in the interface interface region of the cell cycle, and the light chain in the metaphase stage. So the chromomere, centromere regions, knobs also take up dark staining, of which centromeric regions and knobs are the true heterochromatin. In the chromosome, all the centromere fuse to form the long heterochromatic mass. The centromere part is mainly the non-heterochromatic. This is the basically the heterochromatic. So euchromatin is uncoiled and active part. This you need to remember. Euchromatin is not that much condensed. They are lightly condensed. They are the most active part where mainly transcription takes place for the production of different kinds of active RNA and the proteins. So, and the heterochromatin remain condensed and inactive during the interphase portion of the cell cycle. So, eukaryotic chromosome demonstrate complex organization characterized by repetitive DNA. We are going to study this repetitive DNA in our next class, but repetitive DNA, what is repetitive DNA? Is a sequence of repeated many times within the eukaryotic chromosome. They are repeated many times. So like the telomeric region is a part of the repetitive DNA. So if we see the chart of a repetitive DNA, we can divide it into the highly repetitive or the middle repetitive. So highly repetitive portion is the satellite DNA. This is the highly repetitive DNA. And middle repetitive DNA again consists of tandem repeats or the interphases, interspread retrotransposons. Tandem repeats calls the multi-copy genes, which includes rRNA gene, or the mini satellites, which include VNTR, or the microsatellites, which include STR. We will discuss all these things in our next classes. So, and the interspread includes the sign and line, and that is also a very important but an interesting topic, and we will discuss it. And now, if we come to the satellite DNA, which is a highly repetitive DNA and and it is a highly repetitive DNA and is the highly consisting of short sequence repeated in a large number of times. So it's a very short. These are the satellite part of the DNA. Dark in when it is skin, it is actually looking dark in color. So it carries a variable AT reach repeat unit, often forms array up to 100 MB. The monomer length of satellite DNA sequences range from 150 to 400 base pair in the majority of plants and animals. So satellite DNA sequences are mainly located in the heterochromatic region. That is why this is the chromosome structure. This is a chromosome structure, and if we and in this chromosome structure, these heterochromatic regions are actually dark in color, dark in stain. So the satellite DNA sequences are basically located at the heterochromatic regions, so which are mostly found in the centromeric and the subtelomeric region. So they are mainly present in the centromeric or the subtelomeric region, which are regions in the chromosome, but also in the intercalary position. So satellite repeats in eukaryotic are likely involved in sequence-specific interactions. So they are mainly basically involved in the sequence-specific interactions and subsequently in epigenetic process. So we will discuss the satellite DNA later in our different classes. So now we come to the telomeric DNA. So what is telomeric DNA? The telomeric DNA or telomere is a nucleoprotein complex. It's a nucleoprotein complex. This is the telomere present in the end of the telomere of chromatid R. So it is essential for the maintaining of integrity of the genome. It is not a linear structure for much of the cell cycle. Telomeric DNA is basically maintained in a loop structure, which actually serves to protect the vulnerable ends of chromosome. So many of the key proteins in the telomere have been identified, although their interplay So interplay means that they are what is their activity or what is their actual role is still imperfectly understood, and there is a scope of study. So telomeric DNA itself comprises of simple one in which repeat. One in which repeats are present in the telomeric DNA, but in the satellite DNA, the 80 repeats are basically present. So we will discuss these things in our different classes. So now, before the conclude of this class, we will need to understand the function of the chromatin. So the function of the chromatin is to carry out the genetic information from one generation to another by encoding the past history and the future prospects of the cell. So DNA being the only permanent component of the chromatin, it is the sole genetic material of eukaryotes. It never leaves the cell, thus maintaining the heredity of the cell. That is why we call DNA as a hereditary material. This is the most compact region. This is the most compact structure. This is the most important structure within the cell, which actually carries all the information, all the phenotypic information which a particular cell express that actually stored in the DNA, in which is as a genotype in the DNA in the form of genotype, and that the actual form is the chromatin structure. So this is the chromatin structure. Okay, so this is the end of our today's class, and in the next class we will discuss in a very thorough way, the different repeats which is present in the DNA structure. Thank you.